We're going to look and see at what happens in each of these phases in a little more detail in eukaryotic cells. So we're looking at asexual reproduction, and we're going to go through the cell cycle. So remember, the biggest or longest phase is interphase, and interphase is divided into three different parts: G1, S, and G2. You can think of the G's as being the growth phase and G1 being like the first growth phase. This is where it gets bigger, the cell, and it makes things, the organelles and cytoplasm and things inside. S stands for DNA synthesis, or making DNA copies. And then finally, G2 is the second growth phase, and this is where it's preparing for cell division. And we go straight from interphase into cell division, which is divided into two parts: mitosis and cytokinesis. And I'm going to show you some pictures and then, or an animation, and then we'll come back and look at these. And so you'll be able to do this, look at this before your next activity and go through it yourself. So I'm just going to go through it once all the way through. So remember, mitosis is division of the nucleus. So focus on this nucleus here. So in order for the nucleus to divide, we first have to, and make a copy of itself, we first have to make a copy of the DNA. Then the nucleus will have to dissolve. Notice the chromosomes will go to opposite sides. So let me just go through it a little bit at a time. So we're just going to go through it one step at a time. So the first thing that happens is the chromosomes, or the DNA, is going to condense. And so it's going to turn into those X's where it made copies of itself. The outside envelope, or the outside cover of the nucleus, is going to disappear. Now, there are these little things called spindle fibers. They're kind of like fishing lines. One from each side is going to attach to the center, and it's going to be like tug of war. When you have two teams that are equally strong, it's going to pull all of the X's, or the chromosomes, in the middle. And so we go from the pairs condensing to the pairs lining up in the middle. And remember that centromere, that middle portion here, it is the last place that the two identical copies are attached. So we're in the middle during metaphase. And now that centromere breaks, and each identical copy is going to be pulled to opposite ends as the spindle fibers shorten. So we're now getting to anaphase. The A is for moving away from the center. Notice that the cell itself is starting to pinch in. And because this is an animal cell, it doesn't have a cell wall. We'll also see what's called a cleavage furrow happen simultaneously. So we're moving into telophase. At telophase, the chromosomes are at the two ends. We will also see them pinching in. We'll see them start to unravel or become less distinct. And we'll see the nuclear envelope start to reform around either end. And you can see that this cleavage furrow, or the cell, starting to pinch in until we have two separate cells at the end of cytokinesis. Now we'll go through this one more time in real time, and you can actually see a microscopic, a picture under with the video microscope up here. I'll just let it go through. And like I said, you'll have access to this. So you'll be able to see it on your own speed. And I want to talk about what could possibly go wrong during this. And there's so many different things. But one of the things that could happen is something that's called non-disjunction. And so we know a junction is where two things meet. Dis would be where they're not meeting, but non-disjunction would be where they're not meeting. So during the phase where they're supposed to be pulling apart right here, sometimes something happens and they don't pull apart from one going one way and one going the other. Sometimes both of them go to one end. And when that happens, we call it non-disjunction. And as a result of that, I'm not sure if you can see this here at the bottom, but sometimes we have an extra chromosome. So instead of 2, we have 3, and that's called trisomy. Sometimes we only have 1 chromosome instead of 2, and so it's called monosomy. We can see that, and this is one of the reasons they do like an amniocentesis before a baby's born if they think something might be wrong. They can also look at chromosomal abnormalities where parts of the chromosome may become deleted, or they may have an extra part duplicated. Sometimes when they break off, they can